This is mostly the physiological type, not the pathological one. But in some instances, the apoptosis also occurring some pathological type. So before going to the different mechanism and different what are the occasions where the apoptosis are occurring, we have to know what is apoptosis and the definition. So definition wise, apoptosis is genetically programmed cell death. Genetically programmed cell death. This line is totality of apoptosis. Basically, this line is telling everything about the apoptosis. Genetically programmed. The genetic machinery within the cell is within the nucleus. They are focused. This is genetically determined. But in some instances, that could be pathological. So this is tightly controlled. Your organelles are reacting with it. So the apoptosis, the most important point is cell shrinkage is the important clue that the cell is dying, and it is an important pathological condensation. The chromatin, the nucleus chromatin will be condensed, will be intact. Intact cell membrane and condensed chromatin. So, if the cell membrane is intact, is the organelle can come out of the cell? It cannot. So there will be no secondary information. So basically, there will be shell shrinkage, condensation of chromatin, and the plasma membrane is intact. So there will be no secondary inflammation. So these are the main differentiating factors from necrosis to apoptosis. Histopathologically, in apoptosis there will be shell shrinkage, chromatin condensation, intact plasma membrane, and another important factor is usually in case of apoptosis, one particular single cell is dying. But in case of necrosis, a group of cells, that is tissue, is dying. In necrosis, a tissue is dying, like in coagulative necrosis, or liquefactive necrosis, or fat necrosis, occurring in a whole organ, like in pancreatitis, or caseating necrosis in lung. Whole organ, the lung, has major portion of lung is converted into granulomatous inflammation. So our whole tissue is dying in case of necrosis, but usually in case of apoptosis, scattered single cells, that could be single cells, that could be clustering of single cells, or that could be scattering. এখানে ওখানে ছড়িয়ে আছে এবং সেই সিঙ্গেল সেলগুলো মারা যাচ্ছে। সো মনে থাকে, বুঝতে পারছো? সো these are the differentiating features of necrosis from apoptosis. Are you clear about it? কোনো confusion? So basically there is an important mechanism, genetically programmed mechanism for the apoptosis. There are three phases by which a cell can die. Okay. One is initiation phase. Second is execution phase, and third is what? Degradation phase. Initiation means there is initiation of the whole mechanism, and execution means what is initiating should be executed. And these two phases are tightly controlled by a group of enzymes known as caspase. These two phases are tightly controlled by a group of enzymes known as caspases. Okay, and degradation is not controlled, it is carried forward by macrophages. Genetic material. Okay, so there are two set of genes. whether that gene is actually, I should rather be called it a family of genes. This family of genes either could facilitate or could inhibit. So if the that is augmenting the apoptosis known as pre-apoptotic genes. Pre-apoptotic genes. If the family of genes are inhibiting the apoptosis feature, these are known as anti-apoptotic genes. Okay? What are the pre-apoptotic genes? Means these genes are facilitating the apoptosis. What are the examples? Bats, bat, beet, puma, p53. Okay? Bats, bat, beet, puma, p53. Among them, the p53 is very important. You know, but it is known as the Gaussian gene of cell, and it is tumor suppressor gene. Other bats, bat, beet, puma, these are actually pro-apoptotic factors. These family of genes are inhibiting the apoptosis. These are augmenting the apoptosis. But anti-apoptotic factors are BCL2 family genes, that is the BCL2, MCL-XL, MCL-XL, and MCL1. BCL2, BCL-XL, another is MCL-XL and MCL1. So these are the genes which are inhibiting the apoptosis. It is clear? So how these are tightly controlled mechanisms. So how the initiation of apoptosis are occurring. There are two pathways by which the apoptosis can start. Two pathways. There will be two pathways. Either that extrinsic pathway, which is another name of extrinsic pathway is Fas ligand pathway, and another is intrinsic pathway. Why it is calling extrinsic? Why it is calling intrinsic? Extrinsic means there will be something, some injury outside the cell. There is an injury. Mostly these are basically pathological type of apoptosis. So there will be some kind of injury or some kind of response which is outside the cell. And against that response, there will be activation of Fas. Fas binds with the Fas ligand. কি হচ্ছে দেখো? Fas present in the cell membrane. These are basically monomers. They will cross-link with each other. These monomers will cross-link with each other. There are 3 Fas proteins which cross-link with each other, and they bind with Fas ligand. Okay, they bind with Fas ligand. In the next step, this Fas and Fas ligand combination ultimately bind with some death domain, which is known as Fas associated death domain, FAD. This is present in the mitochondrial membrane. Fas associated death domain present in the inner mitochondrial membrane. So this Fas polymerized 3 monomer of Fas polymerized with each other by cross-linking, and they bind with the Fas ligand. These Fas ligand binds with the death domain known as Fas associated death domain, which is present in the mitochondria, which ultimately activate caspase. In case of extrinsic pathway, either caspase 8 or caspase 10 is activated. This is the multiple Fas in the downstream. So ultimately there will be activation of caspases, which will ultimately causes the activation of extrinsic pathway, so that in terms these caspases will initiate the execution. This is known as extrinsic phase, where the injury or the signal is coming outside the cell. এইটুকু ক্লিয়ার হয়েছে না? It's tough. এরপরে আমি যা বলবো সেটা খুবই কি বলবো মলিকুলার লেভেলে and it's really tough for me. So এইটুকু ক্লিয়ার হয়েছে খুব বেসিক কথা মনে রাখো I know there is so many things that written up in province and you cannot understand in this level. Practically speaking, you should remember these key points and you have to utter these key points in front of examiner or write in the exam. Okay? So basically it's an extrinsic pathway which is known as fast ligand mediated pathway where there is cross-linking of fast and 3 fast monomer cross-link with each other and come together to bind with the fast ligand and this fast ligand actually by activation of caspase 8 in case of human this is caspase 10. So in the after activation of caspase 8 or 10 there will be activation of so many caspases in downstream. So the whole thing will initiate the execution process. Another type of extrinsic pathway, another type of initiation process is intrinsic pathway. Why it is known as intrinsic pathway? Because there is apoptotic signal coming inside the cell. There is no external signal, there is internal signal. The cell itself, this is typically suicide. The cell itself is going to die. And there will be two set of genes, pre-apoptotic and anti-apoptotic. When there is activation of it, cell তখন হতে চাইছে খুব simple খুব মানে easy thing. Augmentation of apoptosis by which mechanism? Activation of pre-apoptotic. They are pushing the cell that cell should be dead. Okay, and there will be decrease of level of activation of anti-apoptotic cell. So this balance of pre-apoptotic and anti-apoptotic shift clear each and every one. আমি প্রত্যেককে চাইছি যে এটা মাথা পড়বে না. This is a very basic thing. একদম এইখানে বসেই ক্লিয়ার করে নাও. So there is a balance between pre and anti-apoptotic here, and anti-apoptotic genes are actually non-activation of anti-apoptotic genes or occurrence is no external signal. Okay, so when the apoptotic signal is coming from the cell, within the cell, they are going to they are going to leakage of cytochrome C from inner membrane of mitochondria. Cytochrome C is a part of respiratory chain, pathology point of view থেকে তোমরা জানো. They are leaking of this mitochondrial membrane. Binds with cytochrome. This is a ligand for the cytochrome C. So leakage of cytochrome C. This is the ligand for the. They actually activate the caspases. First caspase which is activated here is same as I told earlier that in extrinsic pathway, like after the activation of caspases 8 or 10 there, after the activation of caspase 9, the initiation of. So there are two types of initiation phase. One is extrinsic, one is intrinsic. In case of intrinsic, the shifting of balance is going to shift to that cell should die. There will be increase in the কি হচ্ছে তোমার pre-apoptotic factors and pre-apoptotic genes are down regulated and anti-apoptotic factors are down regulated, genes are down regulated, so the proteins are decreased in cell and pre-apoptotic genes are up regulated and activation of some molecules which causing the activation of some signals. These signals, there are so many intrinsic signals. তোমাদের এত ভিতরে জানতেই চেষ্টা হবে না. So these signals causing the release or leakage of cytochrome C from inner mitochondrial membrane into cytosol. Within the cytosol, they bind with the APA1 ligand, and this, after the binding of cytochrome C and APA1, there is activation of the protein known as caspase 9, and in the downstream there will be several caspase activation, and this initiates the intrinsic type of initiation phase of apoptosis. Okay. So next is execution phase. Execution phase is basically controlled by the caspases 3 and caspases 6. These caspases, either this activation of 9 caspases, activated caspases 3 and caspases 6, causing the disruption of the chromatin, and actually both. There will be disruption of chromatin as well as there will be disruption of cytoskeleton. Cellular nucleus disruption হচ্ছে, disruption of nuclear chromatin as well as disruption of cytoskeleton. And as a result, there will be formation of some apoptotic bodies. The whole cell is dying. The cell is condensed. Why? Because there is disruption of the nuclear chromatin. So the fragmented nuclear chromatin is congested to a very thin material, and after the disruption of the cytoskeleton, all the organelles are dead, and these things go into a whole cell is going into a formation of thing which is non-functioning thing. It is not a cell anymore. This is a non-functioning thing known as apoptotic body. These things are nutshell of execution phase. So in the degradation phase, the cell is dead. The dead cell is converted into an apoptotic body, and there is some marker of very small condensed apoptotic body. There is some marker. This marker is recognized by adjacent macrophages. প্রথম ক্লাসে বলেছিলাম বডিতে যে কোনো জায়গায় কোনো সেলের যদি ডেথ হয়, the cell will be cleared up. The dead cell, they will be cleared up by macrophage. Okay, like this, like everything here also cleared up. So you have to remember this marker. কি সেই মার্কার যেখান থেকে apoptotic cell কে চেনা যায়? How can I recognize? How can setting is a component of mitochondria membrane, and in the mitochondrial membrane. But when the cell is underwent apoptotic mechanism, the cell is going to phase of apoptotic body membrane to outer mitochondrial membrane. শুধু চেঞ্জ হচ্ছে একটা কম্পোনেন্টের, it's an important integral component of membrane which is present basically in the inner mitochondrial membrane, but there is shifting of place, shifting of position. In fact, it is known as shifting of niche of inner mitochondrial phosphatidyl serine to flip. This is known as phosphatidyl serine flip. This flipping is the marker of apoptosis. We have developed some antibody to this phosphatidyl serine, which is present in outside that is outer membrane, known as annexin 5. Okay, and of course the cell membrane is intact and there is no inflammation. So how can I recognize that apoptosis is taking place? Very dense, small eosinophilic material. There is no basophilic material. Means there is almost এতই কনজেস্টেড হয়ে গিয়েছে, ব্ল্যাক হোলের মতন এতই কনজেস্টেড হয়ে গিয়েছে জিনিসটা, মানে নিউক্লিয়ার মেমব্রেন, that there is no basophilia. It's completely damaged eosinophilic organelles within the apoptotic body. Okay, and there is no inflammation. So of course, if there is no inflammation, there is no adjacent neutrophils and other inflammatory cells. It is of course very difficult to identify in a apoptotic body and under light microscope. Next, in electron microscope, since the shell shrinkage, chromatic condensation, and in some cases there will be some cytoplasmic breaks. Why? Because there will be swell up, the integrity of the cell membrane is lost, so some blade formation, etc. can occur, of course. And third, and lastly, there will be phagocytosis of macrophages. And there will be protein cleavage due to the activation of the cascases and activation of the DNAs causing the damage to the chromatin, and there is fragmentation of the chromatin material by the DNAs. But the most important is there will be in page. It is a chromatographic method. Page. Polyacrylamide gel electrophoresis. Biology polyacrylamide gel electrophoresis. There will be visible DNA ladder pattern. মানে কি যদি সেলগুলোকে tissue where the apoptosis is occurring, the DNA material will be staggered in a step-like pattern. Why? Because simply, first we বলেছিলাম the apoptosis is occurring as some group of cells scattered, and that should be scattered. Some group, sorry, single cell, and that should be scattered. So suppose this is a tissue. Here this group of cell is dead. This group of cell is dead, and there is a distance. এই যে মাঝখানে যে টিস্যুটা আছে, they are not dead, but this cluster of cells are dead. These cluster cells are dead, and this dead cells material, that is DNA, are released, and they are giving this ladder pattern. This dead cell, this dead DNA, this DNA material from a particular tissue is staggered or scattered from each other. So this DNA gives the DNA ladder pattern. But if I do this same thing in case of necrosis, তাহলে কি দেখতে পাবো? A plain DNA plain pattern. There will be no ladder pattern. In case of necrosis, what is occurring? A total tissue is damaged. So all the tissue DNA are damaged. So these are all taken up by the chromatography. So there will be no such ladder pattern is there. A plain, completely clustered pattern is seen. Clustered plain pattern is seen. But in case of apoptosis, there will be staggering of DNA material, which is known as the DNA ladder pattern. এটা তোমাদের এখানে আসবে না, তোমরা যখন 4-5 বছর পর বড় হবে আর তারপর নিট পিক দেবে তখন এটা চলে আসবে। These are things where you should concentrate in the apoptosis basically, and it's of course bread and butter system for you. In the next part, I will cover up some important topics which are not so really important for you, but these are very interesting. 12 বছরের বাচ্চা, okay, Jews, Ascanages stripe in Jews, Persiaতে known as Jews, and there is a special type is Ascanages. Why? Because আমি কেন এই কথাটা বলছি, groups of disease are usually seen in case of these Jews and some tribes of this locality. Okay, there. What we are seeing, they have a very slender and lamba. Okay, short stature, slender plane but short stature. Microcephaly, the head size is small, cytoplasmic, and the pigmentation, there is hypopigmentation, scattered hypopigmentation among the faces and also the leaves. Okay. And in the next slide, what are we finding? Alopecia, scleroderma, light skin, thickened, tough, rough skin, okay, not sweating enough, and the next progeria. What are the similarities between Coombe syndrome and progeria? These are advanced aging. advanced aging syndromes are known as progeroid disease. Progeroid disease. So this is advanced aging as well as known as premature aging. Premature aging. Ages of Dutch death. Why? So we have to understand the mechanism of aging before I go into this mechanism of premature aging. So aspect topic is aging. And I will cover up some other important recent advances of problems. I think these are not so important MBBS question. Aging ta ashbe, but je recent advance gulo bolbo, autophagy ta ashbe, but recent advance gulo bolbo, these are very very important for your NEET. প্রত্যেক বছর এখান থেকে একটা করে কোশ্চেন আসে। আগের বছরে ফেরোটক্সিস এসেছিল, তার একটা বছর আগে আসেনি, তার আগের বছরে কি শিখবো? ডেফিনিশন অফ এজিং, থিওরিজ রিলেটেড টু এজিং, মেকানিজম অফ সেলুলার এজিং এন্ড রিসেন্ট অ্যাডভান্সেস অফ অটোফেজি, নেক্রোপ্টোসিস, পাইরোপ্টোসিস এন্ড ফেরোটক্সিস। So what is aging? Aging is a progressive decline of cellular function and viability caused by genetic abnormality and accumulation of cellular and molecular damage due to effect of exposure of external or exogenous influences. কি পাচ্ছি এত বড় ডেফিনিশন যেটা প্রবলেম থেকে ডাইরেক্টলি না। There are some key features of definition. Progressive decline. It's not a sudden decline. Progressive decline. Some factors. Factors की genetic abnormality and accumulation of cellular and molecular damage. Why this factor? So three things are occurring in case of aging. Aging of cell function due to what? Due to some genetic abnormality caused by accumulation of cellular and morphological changes imposed by or influenced by some external progressive decline of cell due to some accumulation of genetic abnormalities, which is caused by accumulation of cellular and morphological changes, which is influenced by external activities or external factors. So, but this aging is related to, of course, gene. But the most important and interesting fact of this, those genes which are controlling the aging system of each and every conserved is usually conserved. মানে হচ্ছে ইভলিউশন, তোমাদের বায়োলজি ইকোলজি পড়েছিলেন। ইভলিউশন শুরু হয়েছিল একটা ইস্ট সেল থেকে, আজকে আমরা ম্যামিলিয়ানের হোমো সাপিয়েন্স সাপিয়েন্স। Some set of genes remains as same. So these those kind of genes are basically controlling the cell. Theoretical concept. Aging is a very theoretical. আমরা এখন aging সম্পর্কে almost 1% ও জানি না। So there are so many theories proposed to classify or define or actually help to describe the mechanism of aging. What are the theories? Evolutionary theories, decline theory, and cross-linking theory. Just কয়েকটা করে পয়েন্ট বলবো প্রত্যেকটা, বাট তোমাদের জন্য বেশি এটা ইম্পর্টেন্ট। তোমাদের যেটা জানতেই হবে what are the basic mechanism of aging. Basic mechanism এই চারটে লিখছি না, একদম পয়েন্টেই বলছি চারটে। One is DNA damage. Of course, there will be some kind of DNA damage. Now we will cell pathway. Second is cellular senescence. What do you mean by cellular senescence? This is a very important short question. Cellular senescence. What do you mean by cellular senescence? Third is defective protein homeostasis. Normal protein metabolism within the cell, within the within the cell, of course within the cell, but within the body. The normal metabolism of the protein is defective protein homeostasis. And fourth point is deregulated or dysregulated nutrient sensing, means some factors which are maintaining the normal physiological process of nutrition are altered in case of aging. Description আমি আসবো literally meaning basically DNA and genetic theories এর মধ্যেই পড়ছে এই মেকানিজম। বেসিক্যালি DNA and genetic theories এর মধ্যে এই মলিকুলার মেকানিজম গুলো পড়ছে। এই চারটি পয়েন্ট, জাস্ট এই পয়েন্টগুলো শুধু ভাবতে। We are just concentrating much more in DNA and genetic theories, and within the DNA and genetic theories there are four types of mechanisms of cellular division. First is DNA damage. So cumulatively, if there is any metabolic, and there are two types of DNA within the cell, one is nuclear DNA, one is mitochondrial DNA. So cumulative effect of damaged DNA is going to and responsible mechanism for the aging. Usually কি করেছিলাম আমরা বায়োলজিতে বা তোমাদের তো ফার্স্ট ইয়ার ক্লাস হয়ে গেছে, তোমাদের বায়োকেমিস্ট্রিতে আমরা করেছো, if there is a segment of DNA is damaged, they are repaired by DNA mechanism, DNA repair mechanism. So if but our DNA repair mechanism machinery is fully, if it is fully, still it is fully active, some sort of DNA still cannot revive back its to its normal position before dying. Suppose there is an injury to the DNA, and as a consequence there will be mechanism is applied within the damaged DNA. Can the DNA revive back to its totally normal position before, as like before? No, it cannot revive back. Some damage is always present even after the activation of the DNA repair mechanism, even after the repair of the DNA by DNA repair mechanism, DNA repair enzymes. So that in that damage cumulatively causing the aging. কিছু কিছু ড্যামেজ প্রত্যেকটা ডিএনএ তে থেকে যাচ্ছে, ইভেন আফটার দ্য অ্যাক্টিভেশন অফ দ্য ডিএনএ রিপেয়ার মেকানিজম দে আর কজিং দ্য ডিএনএ ড্যামেজ। এখানে মাঝখানে একটা স্লাইড দিয়েছি, এখানে বলেছিলাম দেখো যে থিওরি মধ্যে একটা হচ্ছে কি ফ্রি রেডিক্যাল ইনজুরি। সেখানে this theory is proposed by the Denham Harman. He is telling, কি বক্তব্য স্যার is telling that free radicals and free electrons are produced during the normal respiratory cycle of mitochondria. Right? There is derivation of free oxygen radical, hydroxyl ion, etc., etc. These free radicals and free oxygen or free radicals or free electrons are causing the damage to the mitochondrial membrane as well as the cellular membrane, and cumulatively this damage actually causing the aging. How can I revive back from the free radical injury? There are some scavenger of these free radicals and free electrons. What are those scavengers? These are known as antioxidants. What are the antioxidants? Beta carotene, vitamin C. We can protect the cell from oxidation material, oxidation injury, oxidative stress. That is why they are known as antioxidant. Okay. প্রথমে পড়েছিলাম মেকানিজমের মধ্যে ডিএনএ ড্যামেজ, বুঝলে? সেকেন্ড ইজ সেলুলার সেনেসেন্স। হোয়াট ইজ সেলুলার সেনেসেন্স? Cellular limited capacity of division. Okay, after that capacity is over, will go in a stage of stationary phase or permanent phase. This is known as cellular senescence. Simply. Okay. Why it is known also a replicative senescence? Because after a limited number of division, the cell is not going to replicate, not going to divide. That is why it is known also a replicative senescence. Okay. Aging is associated with progressive replicative senescence. They can't divide anymore after the senescence. This attainment of replicative senescence is nothing but aging. Attainment of replicative senescence is nothing but one is telomere accretion, and second is activation of the tumor suppressor gene. সহজ বাংলায় এটাকে রেপ্লিকেটিভ সেনেসেন্স বলে। এই প্রসেস অফ রেপ্লিকেটিভ সেনেসেন্স is nothing but aging. কেন রেপ্লিকেটিভ সেনেসেন্স হচ্ছে? There are two mechanisms, one is telomere accretion and another is activation of tumor suppressor gene. Telomere accretion. But before that, we have to know what is telomere. Biology data তোমার পড়ছে। কেউ বলো।
Ending of the chromosome.
Exactly. বলো ভালো করে বলো। Ending of the each chromosome. কি থাকে সেখানে? Centromere তো ending না? It's a middle portion. Not ending, it might be middle portion. এখান থেকে সেলের কনডেনসেশনটা হয়।
Secondary construction.
না, not secondary. একদম এন্ডে থাকবে। ঠিক আছে? কোন এন্ডে থাকবে? 3 প্রাইম আর প্রত্যেকটা ক্রোমাটিনে কটা এন্ড? একটা 3 প্রাইম আর একটা 5 প্রাইম। এটা 3 প্রাইম। আর সেখানে একটা পার্টিকুলার সিকোয়েন্স থাকবে। Yes. সেটা হচ্ছে পার্টিকুলার সিকোয়েন্স থাকবে। It's basically a nucleoprotein, a specialized nucleoprotein which is present end of each chromosome. There is a long array of double stranded or TPA-GGG. Long array. এইটা তোমাদের স্বপ্নটা আসে না? টেলোমিয়ার আর টেলোমারেস। তোমাদের স্বপ্ন আসে। এই জন্য এটা একটু দেখতে পাচ্ছি। Anyway, there is long array of double stranded TPA-GGG repeats. It is present at the G reach 3 prime end. And what is the function of it? It causes the ensuring of complete replication of chromosome. During the chromosome, this is one of the mechanisms is presence of telomeres. Can be by preventing at the time of division is taken away from each other to different pole, I mean lower and upper pole of the cell spindle at mitotic spindle, and during the division those daughter chromosomes are not ligated with each other, they are not recombined with each other end to end at the end to end recombination by the mechanism of telomere. They prevent the end to end recombination of telomere and induce the cellular senescence. How they are preventing the recombination? Because there is a DSB protein present at the end. It is known as chromosomal cap. This chromosomal cap formation causes the end-to-end fusion at the time of division. Okay. এখানে থাকবে each and every end some একটা double stranded DNA recombination. Suppose our chromosome is replicated, but each and every chromosome after a particular, okay, but there is a constant length present within a chromosome. Up is that length of the chromosome up to which a cell can shorten, because after the limitation of Hayflick limit, if the cell shorten, cell will not replicate anymore. এটুকু বুঝেছো? Okay. So a certain point of Hayflick limit, the growth is arrested. How the growth is arrested? By some mechanism, activation of some pathway. These are tumor suppressor gene basically. Retinoblastoma gene controlling the arrest of cell when the cell is crossing the limit of. So if mechanism is going to inactive, মানে growth arrest is not going to happen, means this tumor suppression gene is মানে inactivation of means the cell is going to replicate beyond that Hayflick limit, and if there is replication beyond the Hayflick limit means there will be no process of stoppage of replication. The cell is going to replicate again and again and again. Unstoppable replication. That will cause the formation of tumor. Uncontrolled growth. Uncoordinated, uncontrolled growth. Localized growth is known as tumor. So if this mechanism is inactive, there will be telomere attrition and loss. It will cause the genomic instability. And there is number of damaged material, damaged chromosome are closing within the cell. মানে Hayflick লিমিটটা কেন পড়েছে, সিম্পলি বলছি Hayflick লিমিটটা কেন পড়েছে, up to that Hayflick limit there will be some damage, the cell will die or be staggered, be on a particular phase already আছে, but cell যদি তারপরেও অ্যাক্টিভ হয়, মানে তার রেপ্লিকেশন যদি অ্যাক্টিভ হয় তাহলে হয় জিনোমিক ইনস্টেবিলিটি, এটাকে বলা হয় জিনোমিক ইনস্টেবিলিটি। Okay. And what is the role of telomeres? Cause the production of telomeres. By how? By which mechanism? It is at DNA polymerase. So they joins some nucleoprotein at the 3' end of the DNA strand. So the Hayflick limit is not it is maintained by telomere. So by activation of the telomerase, the Hayflick limit can be active each and every. So telomerase is caused যদি একটা সেলের মধ্যে telomerase অ্যাক্টিভ হয় তাহলে তার নাম্বার অফ ডিভিশন বেড়ে যাচ্ছে, কারণ Hayflick লিমিট তো একটা পার্টিকুলার লেভেলেই থেকে যাচ্ছে। Usually telomerase present but in immortalized cell the telomerase is usually reactivated and telomere length is stabilized. So it allows the cell to proliferate indefinitely. And these are all causing genomic instability as well as the tumor. দেখো epithelial renewal হচ্ছে যেখানে at the time of epithelial renewal there is some somatic mutation advanced inflammation which causing the telomere attrition. Telomere attrition causing genomic instability are some important mechanism. তোমরা আরো যখন ক্যান্সার নিউপ্লাসিয়া করবে তখন আরো কমপ্লিটলি বুঝতে পারবে। There will be some accumulation of damage within the chromatin and it causing some genomic instability and it is known as aneuploidy, etc. And unstable manner it will cause some activation of mechanism which causing the invasion of robust telomerase activity causing the invasion of this tumor into the site. Okay. So there is a very important role of telomere and telomerase for the tumor progression. And যেটা আগে বললাম activation of the tumor suppressor gene causing what? There will be এই সেল সাইকেলটা তোমরা মনে আছে কি? G1S G2M. G1S there is a two active in the G1S phase. এইখানে, okay, and they causing the halt or stoppage of tumor cell to enter into the synthesis stage. So if there is inactivation of tumor suppressor gene, the cell will go this checkpoint of G1s into synthesis phase. So there will be replication. So it controls this tumor suppressor gene controls the G1s phase progression, pushes the cell into senescence pathway. If there is activation of if there is activation of this tumor suppressor gene, the cell cannot enter from G1 phase to synthesis phase. Instead of entering to the synthesis phase, the cell go into senescence phase. Third mechanism. So these are all known as cellular senescence. Next, third mechanism is deregulated protein homeostasis. যখন একটা প্রোটিন ড্যামেজ হয় সেই প্রোটিনকে বেসিক্যালি they are folded or they are go into a wastage box by folding. This folding is caused by chaperones. These are anti-stress molecules which causing the folding of proteins. Next, there will be some protein translocation, the normal translocation of they maintain the normal translocation of the proteins, as well as this damaged protein go into a phase of some autosome. কি হচ্ছে বেসিক্যালি আনফোল্ডেড প্রোটিন যেগুলো ড্যামেজ প্রোটিন আছে they are going in a bag known as autosome, and these autosomes bind with the lysosome and formation autolysosome pathway. So if there is damage or uncontrolled chaperones activity, there will be cellular death. Like in case of myopathy, neuropathy, etc. In such cases, this heat shock protein is an example of chaperones. These chaperones are not active. So there will be accumulation of misfolded proteins and there will be cellular damage. And another pathway is insulin and insulin-like growth factor signaling pathway and sarcolemmas. They are basically causing the normal nutrient sensing of the body in an active position. এতটা তোমাদের জাস্ট বুঝতে হবে না, শুধু এইটুকু মনে রাখবে আমাদের বডিতে ইনসুলিন কি কাজ করছে তোমাদের ফিজিওলজি ওয়াইজ দে আর মেটাবলিক অ্যাক্টিভিটি করছে, দে আর লাইপোজেনিক or they active the different metabolic pathway, and insulin like growth factor is an activator of they actually acts as a ligand for the insulin. Okay, so they maintain the normal metabolic activity. And there is a role of sarcolemmas. Sarcolemmas is nothing but sarcolemmas are nothing but the NAD dependent protein deacetylase. They are causing the normal metabolic activity in a very stable position, as well as they reduce the apoptosis and stimulate the protein folding. So if the sarcolemmas is in huge number in body, there will be apoptosis will be reduced and stimulation of the protein folding, and as well as there will be inhibition of the metabolic activity. So basically these things are causing the inhibition of the cellular aging. So if there will be calorie restriction, the sarcolemmas will be active and there will be reduction of signaling of insulin like growth factor 1. This causing the decrease in the metabolic activity as well as normal DNA function repair mechanism will be there. So remember the red wine cause activation of the sarcolemmas and increase the lifespan. And mitochondrial decline theory, another theory. This theory actually proposing that if the mitochondria maintain in a very good phase, the cell will longevity of the cell will be high. Acetyl L-carnitine, it is a transporter of the mitochondrial membrane. If this acetyl L-carnitine hydrogen in a very high level within the mitochondria, it will maintain the normal physiological function of mitochondria. Biological point of view, তোমরা জানো যে there will be glucose is binding with a simple protein by glycosylation. This glycosylation is done by the cross-linking. This cross-linking causing the premature aging, like in senile cataract and tough and leathery skin formation, etc. So these are all about aging.